So back in chapter 4, we covered phylogenetics, specifically cladistics, a method that uses the principle of parsimony to come up with the evolutionary tree that has the fewest number of steps, the fewest number of character state changes. Since homoplasy is a real phenomena in life, that is, similar character states can evolve in different lineages independently, or there can be losses or reversals of character states. That complicates phylogenetic approaches, and the principle of parsimony helps us to find the cladogram with the fewest number of steps. So we reviewed some of that back in chapter 4, and now we're coming back to phylogenetics because we're focused on the use of molecular data in phylogenetics. We just covered gene trees and the concepts of coalescence and coalescence time, and how gene trees don't always match up with species trees because of incomplete lineage sorting. So these are topics that were just reviewed in the first part of this chapter. So now we're moving forward and we're looking at some of the methods of molecular phylogenetics. How do we use molecular data to come up with evolutionary trees, phylogenetic hypotheses? So understanding how alleles arise, which through the process of mutation in various ways, how they spread in populations through reproduction, introgression, etc., allows us to use DNA sequence data to reconstruct phylogenies. That is, we have knowledge and information about genetic data, and that's important for using that data to come up with phylogenies. We can incorporate some of those concepts into our methods. So basically, when we're using molecular data, we are asking the question, which phylogenetic hypothesis best matches the molecular data? Similarly, when we looked at anatomical data, it was the same kind of thing. Which phylogenetic hypothesis best matches the anatomical data? So there are limitations to molecular approaches. Most especially, we can't extract DNA from fossils that are older than 100,000 years. The DNA degrades, and it's just impossible to collect that kind of data. That being said, we have a wealth of living species, extant species. We can extract molecular information from species, from organisms of species, and we're able to use that data to come up with phylogenetic hypotheses that would never have been possible with fossil or anatomical data alone. So while there are limitations to molecular approaches, there's also tremendous breakthroughs in phylogenetics because of molecular data. So phylogenetic analyses use characters and character states. Remember that from chapter 4. But what are the characters and character states when we're using DNA sequences or protein sequences? Well, to answer that, most typically, the characters, say, in DNA sequences is the particular nucleotide position in the DNA. And then the character states would be, well, which of the 4 nitrogenous bases are in that position: adenine, guanine, thymine, or cytosine. When it comes to proteins, then we're looking at a particular amino acid site or position in a polypeptide. Is it the amino acid that's in position number 12? Is it the amino acid that's in position number 213? And then the character states typically are, well, which amino acid is it? There's 20 different amino acids. Which is it? So generally, that's how characters and character states are viewed and used with DNA and protein data. Now, of course, like we talked about in, I guess, chapter 4, there are complications to inferring phylogeny. One of the biggest ones is homoplasy. It's real. Life has evolved similar character states in different lineages. That's convergences and reversals. And that complicates our efforts to figure out the history of life, the phylogeny of life. So these are false signals. The fact that, say, birds have wings and bats have wings, well, that's a false signal. The fact that they both have wings does not imply that they are closely related. The wealth of other kinds of data in anatomy, etc., shows that they evolved wings separately. So homoplasy creates false signals. Homoplasy creates similarities due to convergence. And those similarities are not because of recent ancestry. So keep this in mind. Not only do we have complications coming from what we just talked about earlier in this chapter, coalescence times and incomplete sorting, but we also have complications from homoplasy. So in this sense, molecular data has the same challenges as, say, anatomical data. So there's some background, quite a bit of talking I've done there. There's some background. So some common methods of phylogenetic inference include, I'm going to have a couple slides going over this, maximum parsimony. So similar to what you were introduced to back in chapter 4, except now we're using molecular data. So in maximum parsimony, the topology, that is the tree, the arrangement of the branches, that's a common word that we use in phylogenetics, is a tree's topology, its arrangement of branches. Maximum parsimony says the topology that has the fewest number of changes, the fewest number of character state changes, is the most likely. So a couple components of this, certain regions of the DNA or certain types of mutations can be given different weights, different amounts of consideration as to the importance of that data. So, for example, introns are more prone to homoplasy because introns don't code for anything. And any changes in those sequences of introns would be random changes and would be subject to neutral evolution, genetic drift. And so introns evolve relatively quickly. So what that means is that phylogenetic biologists do not use introns for looking at older lineages that have been around for a long time, because introns change a lot. Instead, we use exons. They tend to be more conserved. They tend to not change as quickly. But if we're looking at more recently evolved lineages, say species that have recently come about, or populations, then introns would be a more appropriate data source because exons are changing too quickly. So we can weight these kinds of things in a maximum parsimony analysis. Another one that you were introduced to back in chapter 4 is consensus methods. Okay, remember that those find the tree, the phylogenetic tree, with agreeing topologies, areas of agreement. So you might end up with some polytomies because the trees disagree in certain areas, but you also end up with resolution and because trees are agreeing on certain arrangements of branches. So go back, review consensus methods if you need to, but they are also used here in molecular phylogenetics. And the last thing that I'm talking about here is bootstrapping, a complicated statistical method for finding levels of confidence in nodes. So how bootstrapping is done is something I don't want to get into here. You certainly can read about it in your book. Very briefly, a random sample of data is taken and a phylogeny is constructed from that data. That data is put back, and then a random sample is taken again and a phylogeny is constructed and put back. And this is done repeatedly. And in the end, all the phylogenetic trees are compared that were done from each sampling of data. And then those phylogenetic trees that have arrangements that are in common, they get a percentage number value put at various nodes to say basically how frequently those nodes were found in common among all the trees. So, for example, a node might have a 70% bootstrap value. So that would be considered a fairly significant bootstrap value that essentially says 70% of the time the trees that were produced from this random sampling of data all came up with these lineages as being closely related. So bootstrapping is another valuable, important tool, statistical method for finding levels of confidence in various nodes. So that's a very long-winded slide, but I put a lot on here, and there's a lot to wade through for sure. So we just covered maximum parsimony, and now we're going to introduce 3 other methods that are used in molecular phylogenetics: neighbor joining, maximum likelihood, and Bayesian methods. And some of these are super complicated. We're not getting into the nitty-gritty. That we would need a course on systematics of just focusing on learning about phylogenetic techniques to really get into this. So neighbor joining. Neighbor joining is what's referred to as a distance matrix method. And fundamentally, it groups taxa, species, whatever you're analyzing to see who's related to who, based on their overall similarity or differences. So if 2 species are found to share the greatest overall amount of genetic similarity compared to what they share with other species, they would be grouped together as more closely related. Species that have more differences between them would be placed farther apart in a phylogenetic tree. And of course, the point here is molecular phylogenetics. So we are grouping species based on their overall similarities or differences in their DNA or protein sequences. And there's an example on the next slide. So we'll show you kind of how this is done after we get through this one. The maximum likelihood methods use knowledge about how DNA and protein sequences evolve. And of course, we know a lot about how they evolve. You guys have taken upper division genetics. You've learned a lot about our genetic material and how it changes. Non-coding versus coding regions, silent versus missense mutations, insertions and deletions. There's different probabilities of these kinds of things happening. Not all mutations are equal. And the point here is that this knowledge, this information is put into the computer program for doing a maximum likelihood analysis. So when somebody does a maximum likelihood analysis and they're writing up their methods in a research paper, they lay out which kinds of parameters they set in their computer program in terms of what was more or less likely in their analysis. So in a maximum likelihood approach, the model of evolution is specified in the computer program. So you put in those parameters of, probabilities of these different kinds of mutations and so on. And then based on what's entered in that computer program, then the possible trees are determined. And for each tree that is calculated, each arrangement of branches that's calculated, then what is given to you at the end of the analysis is a probability of observing the data set based on that tree. And the result that you are given at the end of the analysis is a likelihood value. So the higher the likelihood value for a particular tree or arrangement of branches, then the more likely we would accept that as the hypothesis of how things are related. Bayesian methods determine the probability of a tree, not the probability of observing a particular data set, but the probability of a tree given an evolutionary model and data set. So here we go. Here's the data set. Here's the model that we are using, that we're inputting in the program. So what's the probability of getting this tree based on that data? So it's kind of a reverse approach from the maximum likelihood method. And these are all pretty hardcore statistical methods. And then to end this slide, it's got to be emphasized really strongly that not any one of these methods is particularly better than another. They are different approaches based on what we know in biology about how DNA and proteins change, and they're based on sound statistical methods. What is most often done by biologists when they publish phylogenies is they conduct several different analyses. They'll do a maximum parsimony analysis. They'll do a maximum likelihood analysis. They'll do a Bayesian analysis, and they'll lay out, all the specifics of how they conducted the analyses with the computer programs. And they'll share all the trees. They'll share the most likely, most probable, most parsimonious trees that they got. And they'll discuss the similarities and the differences in the trees, etc. So that's one message I want you to have before we move on, is all of these are methods that are used by biologists, and they're usually all conducted and the results are often compared. All right, so let's take a quick look here at how the neighbor joining method works. So, of course, this is from your textbook. First of all, the analysis, of course, begins with sequence data. And in this case, we're dealing with amino acid sequence data in the beta globin protein. And of course, we can harvest that information from all these different mammals: human, baboon, cow, sheep, mouse, hamster, and chickens thrown in as an outgroup, something more distant. So we begin with that. It could be sequence data for DNA as well, but in this case, it is for the beta globin protein. So in figure A on the top, we've got the sequence for the human, the amino acids written across there by abbreviated letter. And then for the species below that, baboon, cow, etc., if you see a dot, that means they have the same amino acid in the same position. If you see a dash, it means that there was a deletion in that position. And if you see a letter, that tells you that there's been a substitution with a different amino acid. So take a look at that figure A. It's basically the data matrix and or the data set, and that's what's going on. Second step, second bullet, the sequence data is converted to a distance matrix. The distance matrix is basically summarizing the number of amino acid differences by doing pairwise comparisons. So if you look in figure B, you see on the vertical axis, human, baboon, cow, etc. And then going across horizontally on the top, human, again, compared to baboon, cow, sheep, mouse. So the number of differences in amino acids between the human and the baboon is only 2. Between the human and the cow is 6. Between the human and the sheep is 9, etc. Then you go one row down. Well, how about the baboon compared to the other species? And you see the number of differences. So the numbers that you see is the number of amino acid differences when doing these pairwise comparisons. So there's our distance matrix. Then a computer program. Algorithm finds the species with the shortest distances between them and builds a neighbor joining tree. And that's what you see in figure C. Since the human and the baboon only had 2 amino acid differences between them, they are the most similar. They are joined as being most closely related in that tree. Likewise, the other relationships that you see in the tree are all based on overall similarities and differences in the sequence data. So the overall result is the topology. Again, there's that word topology, the arrangement of the branches that minimizes the total branch lengths. And that's what we end up with. That's the neighbor joining method. All right, so after briefly reviewing some phylogenetic methods, showing you how the neighbor joining method works on the previous slide, here's a few case studies showing you how molecular phylogenetics has been used to answer different questions at different levels. So here, we're looking at a population level analysis. So within one species of poison dart frogs in Central South America. So in this case, there was a 3,051 base pair sequence of mitochondrial DNA that was used. And again, remember, mitochondrial DNA is often used for population level studies because it tends to evolve relatively quickly. So choosing the right data source, if you will, is always key. So both maximum likelihood and Bayesian methods were used. I mentioned earlier that frequently multiple methods are used by phylogeneticists, and then their results of each method are compared. And so they use these 2 methods to determine the most likely topology, the arrangement of branches. And there you have it on the right side of the slide, showing you the phylogenetic relationships of these populations based on mitochondrial DNA. Keep in mind, this is really a diagram that's showing the phylogenetic relationships of the mitochondrial DNA. So the mitochondrial DNA may not completely reflect the actual relationships of the populations, but it's being used as an estimator. Now the branch lengths, you can see there's different lengths of these branches represent the amount of divergence. In a sense, it's an estimate of the amount of time since sequences diverged. These are not speciation events. These again are just comparisons of populations and how genetically different these populations have become, really how genetically different these mitochondrial lineages have become. Now the blue numbers on our figure, those are the numbers on the left, represent bootstrap support values from the maximum likelihood model, levels. So in general, from what I remember, any bootstrap values of 70% or 0.7 or higher are generally considered to be fairly rigorous. And you can see there's a lot of values of 1. The green numbers are what are called posterior probability values from the Bayesian analysis. So they're giving the results from both kinds of analyses. And those values are generally pretty high as well. So one might have a pretty high confidence in this mitochondrial DNA phylogeny based on those bootstrap and posterior probability values. Mitochondrial DNA is used a lot in population level studies, and so this is a good example of that. So now let's look at an analysis at a completely different level. Those dart poison frogs were analyzed at the population level using mitochondrial DNA, but here we return back to that question of, well, which fish are most closely related to tetrapods, and how are the various tetrapod lineages related? And so we have a hypothesis from anatomy, including fossils and extant or living species, their anatomy. And these anatomical studies have suggested, and this was reviewed in an earlier chapter, that the lobe-finned fishes are tetrapods' closest relatives. So what does the molecular data say? Well, here we have amino acid sequences for 251 genes. That's a pretty good data set. And a Bayesian analysis was conducted on these data. And there was 100% support at each node in all the cases, except for the elephant armadillo clade. That was just had a support of 45%, which is pretty weak. So one thing to draw out of this is that, yes, indeed, just like the anatomical data suggest, the lungfish is the closest living fish relative to us tetrapods, followed by the coelacanth. So there is consistency there between what the anatomy is telling us and what this genetic data is telling us. And then, of course, we have relationships of various tetrapods, and I can tell you that those relationships are also very consistent with what anatomy tells us. So it's very reassuring when we see a molecular approach in phylogenetics and we see that the results are matching up very well with what more traditional approaches using anatomy have shown us. So there you go. We've seen an example at the population level. Now we're seeing an example for lineages that diverged over, tens, even up to a couple 100 million years ago. Well, hey, let's take a look at our own species, good old Homo sapiens. Molecular phylogenetics has enabled us to address questions that we wouldn't have been able to address without our access to molecular data. So we know from the fossil record that our hominin lineage, the lineage that is the sister taxon to chimpanzees and bonobos, it includes lots of extinct species of human-like creatures, our hominin lineage originated about 6 million years ago, and about 20 species have been identified so far. But how did our own species evolve since coming into being? How has Homo sapiens evolved essentially at the population level? And how might a hypothesis of our evolution explain our distribution around the planet, why we are located in certain places and how we got there? This phylogenetic approach helps us to address those kinds of questions. So the prevailing hypothesis was that Homo sapiens originated in Africa about 200,000 years ago, maybe a bit longer. This is based on fossil data. We got to the Middle East by about 100,000 years ago. We came out of Africa essentially 50,000 years ago, spreading into Europe. So a prediction of this hypothesis is that the molecular phylogeny will show all of our major ethnic groups as being derived from African ancestry. If indeed the out of Africa hypothesis is correct, that humans, Homo sapiens, originated in Africa and then spread out from there, then our molecular phylogeny should reflect that. So 1,327 variable loci were used in this analysis, many of them microsatellites, which is a repetitive source of DNA data that evolves pretty quickly, has a high mutation rate. So microsatellites are very useful for this population level analysis. The neighbor joining method was used to produce this tree. Take a look at the figure. And the primary results that we get from this is that the vast majority of genetic diversity in Homo sapiens resides in Africa. This goes along with African lineages being the oldest within our species. And all non-Africans form a monophyletic group. If you look at near the top of that cladogram or phylogeny, you can see a little arrow pointing out of Africa, the point at which we moved out of Africa, or Homo sapiens did, and moved into, the Americas, Europe, Middle East, into Australia, etc. So, again, molecular phylogenetics is able to shed light on questions of our own origins, of our own species, and how we spread around the world that we otherwise would not have been able to answer. Let's take a look at another example, another case study that is totally applicable and focused on humans, specifically the origin and evolution of HIV, the human immunodeficiency virus. Well, HIV belongs to a group of viruses called lentiviruses, and lentiviruses infect mammals broadly, and typically they invade white blood cells of mammals. Well, molecular phylogenetic studies reveal that HIV is most closely related to SIV, which is simian immunodeficiency virus. So that's found in monkeys and in non-human apes. So we know this. So the question going into this analysis is, when did HIV originate and how did it become transmittable? So to test this, sequences of RNA were used from HIV to construct a phylogeny, which you can see that phylogeny here on the left side of the slide. And the overall point that's being drawn out of this is that HIV-1 jumped from chimpanzees to humans 3 different times. If you check it out in the cladogram, you can see where that happened, color-coded in yellow up on the top of the cladogram. And then in gorillas, it jumped from gorillas to humans twice. So you can see that down on the lower part of the cladogram. So going from SIV in gorillas to HIV in humans. Interestingly, the same mutation happened each time, changing the same amino acid and the same protein. So even though we can detect by the phylogeny that SIV jumped to humans, becoming HIV several times, it actually involved the same amino acid changing, enabling the virus to jump and be able to infect humans. And then our final quick little case study analysis here comes from a box in your chapter. forensic phylogenies, and goes through quite a few specifics there. But, molecular phylogenetics is used in crime cases more frequently than you may realize. Here is a pretty important case that involved someone by the name of Philippe Pédot. And back in 2005, he learned that from his doctor that he was infected with HIV. But despite being given that news and having that knowledge, he went on to have unprotected sex with a number of women who later contracted HIV. So after learning about what he had done, the local district attorney charged him in 2007 with 6 indictments for the crime of aggravated assault with a deadly weapon. And the case relied in part on molecular phylogenetics. So the question was, are the viruses of the infected women most closely related to virus CCO1, which is the variant that Pédot had, or are the viruses that the women were infected with more closely related to other viral strains, more distant or to Pédot? And if you look at this phylogeny, what you can see is that HIV from all 6 women form branches that are nested within Pédot's clade. So the fact that every one of those HIV strains can phylogenetically be traced to have come from Pédot's strain, well, that was convincing enough evidence to give him a 45-year sentence in prison. So what you can see from these case studies is molecular phylogenetics, and we're just looking at the tip of the iceberg here, can be used to answer all kinds of questions, from population level evolutionary questions like with the dark poison frogs to very deep evolutionary questions like with tetrapods, what's going on within our own human species, and forensic phylogenetics. Very powerful stuff.